kind of small. All right, so generally speaking, the nervous system, we've kind of touched on it a little bit throughout the course. It seems like every other lecture I was like, oh, it'll come up from here. Some of the stuff's going to be repetitive, but most part it should be new. So you have a central nervous system and a peripheral nervous system. We've talked about that before. That's pretty much the big nervous system. A lot of it's going to be about breaking it and classifying it into different sections. And we'll take apart each of those sections as we move along. So the central nervous system, as we've talked about before, it's the brain, it's the spinal cord. Okay? So on this guy here, it's just a big blob up here, and then the stuff that runs on the back. Everything else pretty much is the peripheral nervous system. Right? It's the periphery. Okay? Everything that comes out of the spinal cord. Also, the brain has a bunch of cranial nerves that come off of the stem of the brain stem, which we'll talk about later. There's 12 of those in total, and 31 off all the different vertebrae, right between there and the spinal cord. All the little gaps, nerves come out that way as well, outward and around. And those make up your peripheral nervous system, okay? So there's individual nerves, okay? But those nerves they're made of neurons, okay? And we're gonna talk about neurons a lot today. So a neuron is it's not a nerve, but it's a kind of it's basically a cell, okay? A long cell, and in clusters of cells. And when you put those all together, they become a nerve, okay? And then you have clumps of neuronal tissue as well, mostly the cell body of the nerve, which we'll talk about in a minute. And those are called the ganglia, okay? So you have different ganglia throughout your body in different places. A lot of the times they're in the spinal cord, and we'll talk about where in the spinal cord you'll find ganglia and what they look like. Just remember, there's just a bunch of cell bodies, and once you start to break down what a neuron is, that single nerve cell, you'll be able to figure out where the body is, what it looks like, what it does, and all that. Okay, so the classifications is important. So the CNS is the central nervous system. That's just itself, okay? You don't need to classify that any further down. Okay, but the peripheral nervous system has a couple of different classifications. Okay, there's, as you can see, let's go to the chart real quick. I'm not sure what to choose. Okay, so go here. So look at the nervous system as a whole. Okay, so the whole nervous system, right? Then the central nervous system has no breakdown. But the peripheral one, remember everything but the brain and spinal cord, it can be broken down into the autonomic nervous system and the somatic nervous system. Okay? From there, the autonomic nervous system can even be broken down more to parasympathetic and sympathetic. Okay? So this is really important to know. And we're going to talk about each of these in their own lectures for the most part. Today is just kind of a brief overview. But you can talk all about parasympathetic, all about sympathetic on their own.
What is ANS?
Autonomic nervous system. Okay. So the PNS, again, is the somatic nervous system and the autonomic system, right? You can call it these two right here. Okay. And then here's just pretty much the SNS is sensory motor components. Okay. So vision, hearing, taste, and smell stuff. And those are voluntary because those things you can taste stuff, you can hear things, and you can kind of control that. As opposed to the autonomic nervous system, it's also sensory neurons and motor neurons. But you can't really control the autonomic system. Think of it as auto. Okay, so it doesn't happen by itself. Okay, a lot of the time you'll see a lot of the autonomic system in different organs. And so what happens in the ANS, because it's broken down into two, right? Parasympathetic and sympathetic. They both act on that, say, the particular organ, like the lungs, but they act in opposite. So that's good because if you have an effector that does something, and then you're sensing sensing that organ something else, and you change, those two kind of balance each other out. And we'll talk about those in more detail when we get over to that. But that's known as a flight or fight response. So sympathetic nervous system is kind of more for panic. It's going to cause a lot of it's all stress-induced stuff. Parasympathetic is the opposite. It's relax, rest, and digest. I think we covered that early on somewhere. Don't worry about that too much now. We'll talk about those when we talk about those systems separately on their own. But for now, know that this is how it's broken down. CNS is by itself. Peripheral nervous system is broken down into autonomic and somatic. Somatic you can control, okay? Autonomic you can't. And the autonomic can be broken down into the two that balance it out. Parasympathetic and sympathetic. Okay, so what does the nervous system do? It has a bunch of different functions. So you have sensory function, I mean, that's pretty straightforward, right? Touch, taste, smell, all that kind of stuff. It's all done by nerves. We talked a little bit in the skin lectures about, there's a bunch of different nerves in the different layers of the skin, deep down in the epidermis region and the subcutaneous even. There's different sensors for touch, temperature, pressure, and all those types of nerves are all different kinds of nerves as well. Integrator function as well, so the processor, the computer, the brain basically, a lot of your CNS nerves are integrated function type, and they kind of assess what the sensory function or the sensory nerves are doing. And then you have the effector, which is the motor function for the most part. That kind of is, after the computer tells it what to do, then it goes and does it. Okay, so we'll talk about the histology of nerves. Okay, it's really broken down in 3 sections: the neuron, the neuroglia, and the myelination. Okay, those are all the kind of parts of it. But essentially, that's like what a cool nerve looks like. And we'll talk about a nerve in general here. This is a nerve, or a neuron, sorry. Okay, so a neuron is a single cell. All right. The neuron consists of a cell body. It's the big boldy end there. Okay, big circle. That's the cell body. That's pretty much like any of the cells we talked about back there. That's pretty much all the same stuff in it. Okay, so a nerve cell or neuron is specialized because it doesn't look like your typical cell. That looks way different than this guy, right? That's just a circle over there. Okay, it doesn't look like epithelial cell. It doesn't look like osteocytes. Okay, so you can see this thing, the long thing here. We'll talk about in a minute. Definitely you can pick this out of the other types of cells for the most part. So you have a cell body, which has got all your cell function stuff, your nucleus and all the organelles and things, okay? Then at the body here, you have these projections. They're called dendrites, right? And they're used to communicate with other cells and surrounding as well, okay? They kind of pick up on things around them. And you have the long part here, which isn't always long, but it is here in this model cell. It's an axon, okay? And that's when these dendrites sense something at the tips here, then what's going to happen is you're going to need a signal to go to the other end for the effector or whatever is down here. So to carry a signal from here to here, you have this axon, and you're going to have charge go through here. Positive charge, positive and negative electrical charge. And we'll talk about how that happens in a minute, but note that it's going to travel down the axon. Okay? And then here you have the synaptic terminals. You remember from muscle, there was the muscle neuron that came up to the synaptic cleft of the muscle. That neuron was pretty much something like this. Those are the bulbs at the end that have the acetylcholine in them. And then the acetylcholine was released onto the muscle, which had acetylcholine receptors. And that's where reaction potential kind of went down into the T-tubules, released calcium, all that kind of thing. So that all happened down there. We had this big magnified picture of what happens in the synaptic cleft. Those are little bulbs there at the end, okay? Does that make sense? Maybe. If not, ask questions. Okay, so we can classify neurons by structure. We can classify them by function as well. Because there's a bunch of different types the way they look, and they also have different functions. Okay, so let's talk about structure. So you can have multipolar, bipolar, unipolar neurons. Okay, and we'll see in the next picture here. If you have your slides in front of you, see how they look kind of different, the cell bodies in different places and things like that. So let's first talk about multipolar. So you have a bunch of dendrites, okay, and one axon. So it's kind of like this guy here, all these dendrites, okay, and then you have the one axon. That's a multipolar neuron, okay. You have bipolar, where you only have one dendrite, one axon. So you wouldn't have all these guys. You'd have like one, okay. And then you have unipolar, which is just dendrites and a single axon. They're kind of fused together. The axon's not this long thing. Remember I said the axon can vary in length? Well, in a lot of your sensory neurons, you don't have a very long axon, okay? You have several different neurons kind of closer together, and there's not a very long axon there. Yes? Okay. Okay, I'll slow down. So again, structure-wise, 3 different types. Uni, bi, and multi. And from that, you should be able to figure out kind of what's going on. And you can see in the picture from here. So you see the bipolar, The axon, single dendrite. It has projections off of it. There's still one dendrite coming off the cell body. Okay? And then you have the axon. All the synaptic bulbs down there. You see how there's arrows pointing here? That's where if something is sensed here, the action potential which forms over here kind of flows this way. The how do you put it? The message that's picked up here is relayed this way. A unipolar or a pseudo-unipolar, you get the central axon here and a peripheral axon. Okay? The dendrites, multiple bulbs here. Then your multipolar here. Just like the first picture we saw with all the different dendrites, right? And then an axon. Cell body is connected to the dendrites. And we'll be able to see all these different types on the slides as well. So again, it's not necessarily important to know where you find these different types of cells. It's written there, there's a few examples. I think your book talks about a few examples. Just more so about the fact that there are 3 different types. Okay, so we can talk about neurons by function as well. So now we know there are 3 different types as far as structure. So let's talk function. You can have sensory, motor, and then there's also interneurons. Okay, so sensory is kind of your afferent. Sensory. It's just, at fingertips, your touch, taste, smell. Those are all sensing stuff. Those are sensory neurons. And they're known as afferent. Okay, and then there's efferent, which are effectors. Efferent effectors. Those are motor neurons, is what they call it. They're not necessarily doing anything, motor like skeletal stuff, but the effector is known as motor neurons. Okay, and then you have neurons between that too that kind of relay messages, and those are interneurons. Okay, they kind of also integrate the information that's received from the afferent nerve. So the afferent picks up something, looks, senses it, goes to interneurons, which are processing. The interneurons is essentially the computer, and then the effector is the motor neuron on the outside. Okay, so we kind of talked about this. I think it was the first or second lecture. I think it was the first lecture. That whole system. I think it was an exam question on it too, for which the answer was effector. That's how it works, but it's mostly neurons. It's all neuron stuff. It's not any other type of cell. Okay. Does that make sense? Yeah. Okay, so those are the neurons. Then we're going to go on to the next thing, the neuroglia. That's like the glue. It's kind of stuff in between neurons. If in your brain, it's not just full of neurons. There's space in between as well. And there's these neuroglia. There are a bunch of different types of cells. There's about 6 different kinds. They're not neurons though. They're not structured like neurons. They don't really look like neurons, but they have a bunch of functions. They kind of take care of neurons. So they're kind of like their helpers. So especially in the brain, there's a bunch of different types, and the outside peripheral nervous system, there are a few as well. So what do? They hold structure. They hold a framework for all the neurons. They're responsible for repair, and they do what's called myelinate or myelination of these cells. And we'll talk about what that is in a minute. So again, there's 4 types in the CNS, and there's 2 types outside of the periphery. These are the different types. You don't really need to know them in too much detail, but know that there are different types. If someone asks you, like, what's in the astrocyte? You can tell them it's a neuroglia cell. It kind of helps out with neurons, okay? And these cells are a lot of the reason people get different brain tumors. Because there's so many of these, they outnumber neurons by like 10, 50, 200, depending on the region. There's a lot of them. They're constantly replicating, so there's room for error, and that's where a lot of tumors arise from. And all of these have different types of tumors associated with them as well. You have some in the periphery as well, Schwann cells, satellite cells, and so since they're for structure, repair. If you don't have ones that are very competent, your nervous system kind of breaks down too. So there's a lot of pathologies related to these types of cells. If they're not functioning properly, you're going to have a lot of symptoms regarding your nervous system, maybe touch, feel, different things like that. So myelination, basically, what happens, I'll tell you guys now, is when you have your axon, it wraps it. These cells, some of these cells, the different ones like Schwann cells, they're responsible for wrapping the neuron. Okay, they hug it basically, the big axon. And when it does that, it kind of insulates that neuron. Okay, and when we talk about charge being propagated across the neuron here in a minute, you'll come to learn that having those different parts of myelination, those blankets, the charge can jump between those different blankets that are along the axon, as opposed to if there's no blanket, the charge has to go slowly all the way through to the other end. But when you have the blankets, it can jump over those blankets between the spaces in between them. Okay, so that makes the impulse travel quicker down the axon. All right. So if you don't have those Schwann cells working and they can't insulate, your charge doesn't propagate very fast. Something like multiple sclerosis, those cells are kind of not working properly. So that's why people with multiple sclerosis, they're numb, they're not functioning as well, they're slower to react to things and move, and they have difficulty picking up pencils towards the end of the disease. But it has to do with these cells here.
So what causes that?
That's up in the air. There's a lot of different theories as to what can cause something like MS, but if we knew, we'd have a better way to kind of target it right now. But essentially, something going on there. There are things out there that can, help with flare-ups and stuff like that, but I couldn't give you a definite answer exactly what is causing it. There's still a lot of debate about that. Okay, so myelination, the blanketing thing we were talking about. Let's talk about it. So it's for insulation. Okay, so you have the axon here, the long projection, right? And so these cells, these Schwann cells, which are a type of neuroglia, come and they wrap themselves around the axon. You see that? They put themselves around it, make a blanket. Okay, and they're made up mostly of lipids and proteins. Okay, and so those spaces in between are called the nodes of Ranvier. Some French guy, I think. So he found those, discovered them, named them after himself. And so basically, when a charge comes down this way, it has to travel usually all the way through. But when you have these blankets here, the charge can't really go here. But what it does, it pops, it jumps from one tip of one node of Ranvier to the next. So it goes a lot faster. The charge can jump. That's chemistry stuff. We don't really want to get into too much detail. But again, instead of traveling down there nice and slow, it jumps. And it comes a lot faster, okay? So as you can see, this is kind of blown up here. There's a node, you see them there and there and there. And it's wrapped several times, hundreds of times, depending on the type of nerve. So charge conduction down this axon is dictated by two different factors. One being if it's myelinated or not. So if it's not myelinated, is the charge going to go faster or slower? Slower. Okay. So the other thing that dictates how fast charge goes, the chemistry thing, in physics thing, is the diameter of the axon. Bigger diameter, faster spread of charge. Small diameter, they say this is a small diameter, it's going to be slower. So if you have a myelinated, large diameter axon, super fast. If it's not myelinated and really large, it's okay. If it's non-myelinated and a really small diameter axon, really slow, really slow propagation of charge. Okay? So again, two factors, diameter and myelination. Either myelinated or not, and your diameter is either small or big, or somewhere in the middle. So the interesting note here is that myelination increases with age. That's part of the reason why babies are always falling all over the place, because all of their nerves aren't myelinated very well, so they have issues with balance and things. And that's why the learning process of walking and using their, and talking and things like that is control of all of your nervous function. And a lot of that isn't really too apparent as before a kid because they don't have a proper myelination of all their nerves yet. So now if you see a kid somewhere falling over, you can talk about the nerves are not myelinated. And then you'll remember for the exam, and then you'll get it right.
So then what happens with older people that say big babies?
Yeah, your neuroglia kind of peter out after some time. That's one of the things that would happen with the age. But I mean, as far as a specific cell type, like Schwann cells just going debunked, that's something like in a pathology like MS. But yeah, you have some loss of function, and I mean it's a multitude of different things. Everything from the different vitamin levels in your body to the DNA we talked about when the telomeres get cut off, right? The ATADs at the end of the DNA complexes. But myelination, at the tail end of life does kind of come down, and that's not because the sheets just start falling off, but the Schwann cells are starting to kind of tire out. You don't get as much function out of them. And those cells are probably tiring out because, again, with the DNA, you know, cutting off all those ATs at the end of the DNA script, then you have less function of the cell as a whole, right? But it's fair to say, yeah, that you will have some decrease in myelination. Okay, so nervous tissue can be in different bunches in the body. All right, you'll have clusters, which talk about of the cell bodies, and those are ganglions. Okay, you'll find those in different parts of your body, and we'll take apart the spinal cord in one of these models, and we'll look at where the ganglion are. So again, the bundles of axons are a nerve. Okay, so all those neurons together is pretty much a nerve. So when you have, say, like your sciatic nerve or something, there's a bunch of neurons which are nervous cells running together, okay? And they're enclosed in the sheath and things like that. But a nerve itself is like if you split a part of the nerve, you might lose some function. But I mean, if you have the whole part of the nerve still intact, you might still have some function. But if you cut the whole thing, then you'll lose all function to wherever that thing is kind of serviced. But just know that if you have a big tube, which is a nerve, you have a bunch of neurons going through there. Okay, mostly the axons, because that's the long projected part, right? Okay, so gray and white matter, you always hear about, gray matter, white matter in the brain. The brain is mostly like white matter, but it's gray around the outside. So when you cut a cross section, you'll see there's some discrepancy in color, and you can see a pretty defined border. But white matter is myelinated axons, and gray matter is mostly just unmyelinated stuff, and the cell bodies, the dendrites, and everything else. Okay. Okay, so now let's talk about action potentials. Does anybody remember what an action potential is? So you send an action potential, go ahead. No? Okay. So carry a conduction of pulse, right? Basically charge. Like in the muscle cell, we talked about propagation of charge, right? You're carrying information from the dendrites all the way down the axon to the synaptic bulb. Action potential is charge running, okay? So you have a membrane potential, so you have a cell, you have a neuron, right? The neuron has an environment around it too, right? Everything outside of it, everything outside of the cell membrane, right? The lipid bilayer, whatever you want to call it. So there's a charge inside, there's a charge outside of a cell. Isolate one cell in your mind. Think about one single cell, like that neuron with that back here somewhere. Think of this guy here. So it gets a charge inside and a charge outside, okay? So that charge is based on primarily sodium, potassium, both. Because those are ions, right, in solution. Sodium positive, potassium positive, right? So there's a whole ton of potassium inside this guy. And there's a good chunk of sodium outside. So they're not equal. And there's a few other, ions and things that contribute, but think of just those two for now, for this time. So they're not equal. So if you have a bunch of positive charge inside, but you have even more positive charge on the outside, there's a differential there. Okay? And millivolts on a normal cell is like -90 or something. So yes. Go ahead.
Is that why, because I am deficient in potassium, so my whole body will shut down sometimes, and you can't move.
You can't move. Yeah, a lot of issues with potassium. It's going to be a lot of nerve conduction and things. It's a little more complex than that. But for the most part, yeah, you can say that your nerves are not functioning properly because of your potassium imbalances. Yeah. You start to cramp up, you get muscle cramping and things like that. Well, your nerves aren't able to kind of keep your muscles in check and have opposite balance between flexing and extending in something, and so you'll have one conducting more than the other. And that's why they say eat a lot of bananas when you cramp up. Bananas are high in potassium, right?
Did you say that sodium is on the inside and potassium on the outside?
Potassium's in a higher concentration on the inside, and sodium's in a higher concentration on the outside. Okay. So you have a difference there. So usually there's more positive charge on the outside. Okay. And so what that's going to do is if you have that going on, if you can switch those charges, you can send a charge down this way. Let's say something happens over here, you can find a way to get a bunch of sodium to rush in, and then your charge difference is going to change, right? So you'll have a change in charge and that charge can propagate its way down here to the end and cause something to happen. And we'll talk about how that happens or what that exactly is here in a second. Okay. So let's see. That's good. Sorry. Okay. Well, we'll go back to this guy. Okay. Okay, so along this axon, you have different channels, okay? You have sodium channels, potassium channels, right? And those channels are going to allow those ions to move across the membrane, okay? There's two different types of channels we're going to focus on, okay? There's like gated channels and there are leak channels. Leak channels are usually open, there's nothing happens with those. They're just open and they allow sodium, potassium to leak in and out, okay? Mostly potassium, they're more receptive to potassium. So the potassium Ka ions are going to constantly be leaking out of a neuron, okay? And they're in higher concentration on the inside here, so, you know, laws of diffusion, they're going to want to come out of the cell, right? There's some sodium leak channels that come in as well, okay? Then there are voltage-gated channels, the other type. Something needs to happen to that channel for it to open up. Those are primarily sodium channels. There's a lot of sodium on the outside. That sodium wants to come inside because there's not as much sodium inside this cell, okay? And it's also a negative charge on the membrane. There's a difference of membrane, negative 90 or so, negative 70 depending on your textbook. So once those voltage-gated channels open here, that sodium's going to rush in, okay? So you have all this positive charge out here, and not as much in here. When all that positive charge rushes in, that membrane potential is going to go up from negative something like negative 90, it's going to start to climb, okay? And when that starts to climb, all that sodium rushing in, a little bit later, the potassium voltage-gated channels will open, they're slightly delayed, they're going to start to come out of the cell. And that's going to bump the charge even higher, okay? And so when that happens, you have this charge differential. You have this positive, think of it like kind of a positive, like say when you touch someone and you get shocked, think of it as like that shock, okay? The little shock's going to start here. And then that's going to cause the cells right next to it, not the cells, the pumps right next to it to change the conformation as well. And those sodium voltage-gated pumps are going to open too, the channels. And so then they're going to let in sodium. So then that charge that starts right here is going to move this way a little bit too. And all that stuff's going to happen here. And then you get all the charge flowing in here, and the guy next to it's going to go, oh, there's positive charge. He gets shocked. Then that sodium channel opens up. And then you have this charge, and it just kind of, it's like a domino effect. And then this charge then, this positive charge, that little shock, is going to carry its way all the way down to the other end, okay? And once it gets over here, we'll talk about what happens. But note that that's kind of how it propagates along. So there are the types of channels. Again, so leak channels, they're constant stream. It's not much, but they're there, so you should know about them. I mean, they don't contribute as much, but there's always a little bit of leak, okay? Kind of like you get like a dripping faucet. Whereas the gated ones work on command, okay? And that's when the dendrites are going to sense something happen, they're going to tell those channels right at the end there of that neuron, at the axon hillock, that's called, they're going to be the first ones to change conformation, and then that charge is going to start going that way. So again, there's a high potassium inside and high sodium outside, okay? Okay, so again, this is straight out of your book. This is how action potential is generated. It's a sequence of rapidly occurring events that decrease and reverse the membrane potential. And it eventually goes back to normal, and we'll talk about how it resets in a minute. But so basically what happens is you have that depolarization. There's a threshold of that negative 90 value, or negative 70. We'll call it negative 90 for this class. So when that value starts to come up a little bit, it's kind of an all or nothing thing. So when those sodium ions come into the cell, that negative 90 it's resting at, it's going to start to come up, right? So it'll go to negative 80, negative 70, come up towards zero, right? Because if you have a negative charge pool and you're adding positive to it, it's going to come closer to zero. Does that make sense, right? So once you reach the threshold, then it's all or nothing. Like then the action potential is going to happen. Like those gates are going to start opening. The neighbor gates are going to open. The neighbor gates are going to open. The domino thing is going to go on. So you can kind of toy with the domino if you have them all lined up. You can kind of tap it a bit and it'll rock back and forth. But once it hits the threshold and it falls over, all the rest of them are going to go too. Okay. So you have the depolarization, those voltage gates open, sodium rushes in, and then eventually the potassium channels, I think I might have said this opposite earlier, but this is how it is. So basically those potassium channels will open a little later. And so what that does is you have that positive potassium inside the cell leaving the cell. So what happens when you're losing positive charge? It's going to start going back to negative. Okay? So we'll look at that here on this guy. Okay, so you're resting at negative 90. Okay? At time 0, it's just kind of when it's hanging out. So right away you have the action potential start. Okay? It's called the depolarization. That's when all the sodium comes in. Again, making the cell going from negative 90 inside, because say it's like there's a little bit of potassium inside, there's a bunch of sodium on the outside. A little bit of positive, a lot of positive. So that's negative with respect to the outside. The inside of the cell starts negative with respect to the outside. Does that make sense? Right? Okay, so once all that sodium starts coming in, then it's going to go towards 0, right? All those channels open. But then a little bit later, you have what's called repolarization, because this is the polarized area. So it's depolarizing, going back to 0. This polar is like, opposite charges, positive, negative. This is neutral. So when it starts to repolarize, how's that happening? Those sodium gates start to close, okay? So no more sodium's coming in. But at the same time, well, again, those potassium channels open up. And since there's a lot of potassium in the cell, it's going to want to all come out. And when the cell itself is losing that potassium, it's losing positive charge, meaning it's going to go back and be negative. Yes?
Why does this happen?
Because, why does it happen? Well, I touched this. Well, I touched this and it's really hot. Those nerves or the sensory nerves, the afferent nerves, the dendrites sense that change. Well, the rest of my body needs to know about that. The brain needs to know. So think of it as one axon all the way up to my brain, okay? I feel it, dendrites see it, the cell body's here somewhere, and then it causes that change at that first axon neurolike area where the sodium channels change. Okay, so you have that positive charge. Because how can you tell, say something happens here, how am I going to tell my brain? Chemically, you can't just, you need to know somehow. So that charge has to be carried, and that's the way our bodies have kind of been designed, is there'll be a charge that'll carry. Instead of having something happens here, I touch it, it's hot, then there's a chemical change, that chemical gets carried up here, or that chemical turns on another chemical and releases a chemical, and does this, and that. The fastest way is electricity. So electrical charge is like the fastest way. So you can touch here, and that charge goes up, and then at the end of the axon, you have the synaptic bulbs, then you can have something happen, and we'll talk about what that is.
So basically, it's to sense some kind of change.
Yeah, that's what an afferent neuron does. But not just sense change, you're going to have the motor neurons that are the effectors too, right? Well, if the processing neurons in your brain want to change something, well, so you feel something hot here. Brain hears that. The center in your brain that's going to detect heat and cold and things like that, it's going to go to that particular place because that's where you're wired to go. Well, then it's going to have to tell what's going to happen next. What's your brain going to want to do? Yeah, take it away. So then you can have effector neurons that are going to be innervating muscles. Right? So to get that message down to those muscles, it's the same thing. It's going to be the same positive charge thing on a different neuron path. That's why the wiring is so complicated in your body. You have something here that's going to sense, it's going to go to a certain part of your brain, and that's going to tell a different part of your brain, okay, well, tell that guy to tell the muscles to move there. That's the reaction there. But it's just carrying the message, basically. And that's why you have this positive charge kind of thing going on in an axon. So you have to have a way to get it done quickly for the most part. And that's why we're such alert as people when things are on the third, most people. You have to be able to have those charges and stuff quick, like milliseconds. It's very fast. We couldn't really even comprehend how fast it really is, but yeah. So anyway, so all these sodium channels are opening up. The sodium channels are closing, but the potassium channels are opening, coming back down. And sometimes you have hyperpolarization. Because those potassium channels can be kind of slow, like they didn't even open when here, they're like way over here, starting to open. They can kind of close slowly too. And so because of that, you're letting out a little too much potassium. And then you're going to come below that regular 90. But eventually it'll reset because you have other pumps that'll change the balance back. because the cell wants a lot of potassium inside of the cell wants a lot of sodium on the outside, okay? So there are pumps designed, which we don't go into too much detail about, to reset it, okay, to make sure we come back to this level, okay? So at this point here, you have a bunch of potassium outside now, and all the sodium's in the inside. So again, those pumps are going to switch those. Remember we talked about the sodium-potassium pump before? Letting in potassium, letting out sodium. It lets out 3 sodium, and then 2 potassium in. That was one of the first types of carrier proteins we talked about. Well, that kind of pump is going to work here to get more potassium in while it throws out sodium, just to balance it back to over here. Okay? That's an action potential. Okay? So again, the nodes of Ranvier lead to a quicker impulse. So you don't have that long axon, you don't have those pumps all the way along there. They're only at the nodes pretty much that need to work. And that charge can jump along the axon, okay? And since the axon is really long, or at least, it is in the body, if you have an axon that's like this long, that's really long on this chemical level for things to happen. So since that's the case, you can jump, if you can propagate the charge, by jumping, skipping across, that's a lot better and it's faster, okay? And we were talking about the diameter stuff. Okay, so here again, it's kind of more of a time-lapse picture of what's happening. So you have here the axon hillock. That's the name for the part where the cell body joins the beginning of the axon. You have that sodium rushing in, okay? And then you have that positive charge thing. And then remember, the next set of guys are gonna open up because they're right next to it, and those sodium pumps, or the channels, sorry, they're influenced by charge. So once these guys are charged, ready to go, then the guys next door, that charge kind of shocks them a little, and then they change. The guys next to that shock, and then that's how it gets carried on the way over here, okay? And then you have, at the end, what's going to happen here. Okay, so we talked about it kind of in muscles. Remember how you had a charge, we always talk about just what happened at the muscle and the synaptic cleft with the neuron. Well, this is all the stuff leading up to that. So you would have, what happened in the muscle? You had acetylcholine released, right, from the synaptic bulb, and then that went on to the muscle side, to the receptors, right? Well, so you had that charge coming down the axon. It comes to those synaptic bulbs. Now we're going to kind of zoom in on what's going on the other side of the neuron side. At those bulbs, once that charge comes all the way to the end, there's a bunch of calcium pockets, okay? So the axon comes down, down, down. That charge, the action potential, that change in charge comes here. And here, instead of having voltage-gated sodium channels like you've had all the way along here, you have voltage-gated calcium channels, okay? Calcium will then rush into the cell from the outside, okay? Because those gates will open at full. Calcium rushes in, and what happens then, it attaches and signals these synaptic vesicles, these little stacks, right? And those kind of move to the membrane, right to the end of the bulb. And then they dock to a docking protein, and they release the neurotransmitter, okay? So whatever that type of neuron is, those neurotransmitters, there's 1.5 million different kinds. Whatever that neuron specializes for, there'll be a bunch of that neurotransmitter hanging out here. And once that calcium comes in, tells it, hey, go ahead and come to the end here, it'll come out, fuse out, and then it'll bind to the receptor on whatever the postsynaptic cell is. In the case of muscles, it was muscle fiber. But it could be another neuron. It could propagate the signal to a different one. It could be the afferent talking to the inner neuron, which will then do the same thing again to an effector neuron, okay? So that's how one neuron tells the other neuron to do something. So this neuron here is going to get this neurotransmitter, whatever it be, acetylcholine, it could be dopamine, whatever it might be. And then that one's going to get that, and when that happens, there's going to be a chemical change in there, then it's going to start the whole process over. At its cell body, it's going to go to the axon hillock, and those sodium gates are going to open there, and then you'll have another charge sent to the next thing. And eventually it'll end up at something like a muscle, right? And then now you can have pulling away from the heat kind of thing, okay? So that neurotransmitter can run out, right? That's what we talked about before, like if you're lifting a weight or something, you can't just hold on and lift it forever and ever, because eventually you run out of neurotransmitter. One of the components is you run out of the stuff, and then that nerve doesn't get a signal anymore. So again, a bunch of different types of neurotransmitters. Acetylcholine we talked about. You don't need to know these, but have glutamate, aspartate, different things. and they all work in different ways. And that's the end of that.